In this video, we will discuss about the function of these Dsup proteins, whether it is able to protect the DNA from radiations or not. In the previous couple of videos, we have understood the enhanced lipid nanoparticle or polymer lipid nanoparticle transfection method, which can transfect the mRNA with more efficiencies compared to the previous lipid nanoparticle methods. So they use their enhanced lipid nanoparticle methods to transfect these Dsup proteins into the cytoplasm. And since these Dsup proteins do not have any commercial antibodies, what they did is they use the mRNA Dsup or fuse the mRNA Dsup with the EGFP reporters mRNA and control the and check its transfection efficiency with only EGFP mRNA. And what they are able to see is that these Dsup proteins are expressed more into the nucleus. Whenever these Dsup fused EGFPs are expressed, they are expressed into the nucleus, whereas when only EGFP mRNAs are expressed, they are expressed throughout the cells. And the reason is that this Dsup fused mRNA has this NLS sequence, uh, which uh, which which is nuclear localization sequence, which direct the proteins, translated proteins into the nucleus of the cells. That is the reasons with these Dsup mRNA proteins that are expressed are localized into the nucleus, as their job is to protect that DNA damage. So its natural position should be into the nucleus, as DNAs are present into the nucleus. Then they check the impact of these Dsup proteins using the COMET assay, and COMET assay is used to determine the damaged cells. So when they for the control case, they are able to see some single stranded DNA, but when they do not use the Dsup proteins uh, and use the COMET assay, they are able to see more amount of single stranded DNA. But when they use the Dsup mRNAs and do the COMET assay, they are able to see less number of single stranded DNA, and the difference in control and Dsup mRNAs are statistically significant. And this the figure B in the panel A is for the oral epithelial cells, and middle figure of the panel A, and the right hand side of the panel A is for the colorectal epithelial cells. And in this case also they see the same results, that is single stranded DNAs are low in the case of where these mRNAs are transfected, and when they check these damaged cells using the COMET assay. And they also check the reverse thing, that is they check whether Dsup proteins is able to prevent the damage of the prevent the DNA cells or protect DNA cells or not. In order to check that, they use this dye, that is gamma H2AX, which is used to stain the DNA. So in this case, when the cells are transfected with the Dsup mRNAs, they are able to see less signals for this gamma H2AX for both colorectal epithelial cells and oral epithelial cells, indicating that the Dsup proteins Dsup proteins covers this DNA, and thereby prevent any further effect from the other dyes or other external things that is trying to impact this DNA. And this the this staining of this gamma H2AX is shown on the right hand side of the panel B, where we can see that in the case of control case, the signals are higher compared to the case where the Dsup proteins is used. And once validating that it will protect, Dsup proteins is able to protect the DNA, they wanted to check the actual experiment where they use different intensity of the radiations and check whether Dsup proteins is able to prevent this DNA damage through the number of colonies. And what they are able to find out, at different intensity of the radiations, they are able to see that number of colonies are high when the Dsup mRNAs are been transfected into the cells for all the cases that they have tested, that is 4 gyron and 10 gyrons. And in both the cases they are able to see that number of colonies are high when the Dsup transfected, when the cells are transfected with Dsup mRNAs, compared to the case where cells are not transfected with Dsup mRNAs, or the cells are transfected with only GFP mRNAs. So all these results finally points out to finally comes to a conclusion that it's the Dsup proteins are able to cover this DNA and prevent them from the radiations impact, and thereby helps them or protects them from the DNA damage that is caused by the radiations. So I hope you understood the results that are presented in this figure. If you need to understand more about their experiments and their supplemental results, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.